can't, no, that's not, because I'm downloading this. I can ask you to define it. So what is a metabolism? And so you can use different definitions. It's okay, you don't need to have my wording, but we do have to have the right idea about it. Some books will say all the chemical and physical workings of the cell. So Marjorie Cowan had a book that I used for years, and that's her definition of it. So, you know, anything the cell would do, essentially. And that's okay, but shoot, that's so vague, that doesn't make any sense to me. I prefer this definition, and that is the ability to convert energy from one form to another. And so if you consider right now that you're sitting there and moving a pencil, or maybe you're scrolling on your phone, whatever it is that you're doing, you know, that takes energy. Well, what is the energy you're using? It's ATP. Okay, that's fine, but where did you get that? Okay, well, you're breaking something down for that energy. You either are breaking down your breakfast, your lunch, maybe you haven't eaten, and you're breaking down stored energy that you have to provide the ATP to be able to do what you need to do. Okay, and I think we've talked about this already. Like, if you did absolutely nothing and you're like 150 pounds, supposedly that's 2,000 calories just to stay alive, just to pump your heart. Okay, so this ability to convert the energy of the foods that you eat into ATP, that's a metabolism. We say that metabolism and having one is key to being alive. Okay, that's part of the definition of life. All living things have a metabolism. We say that, we agree to it, but we don't normally define it. So I'll give you the example that I know I've given you before. If I have 2 slabs of steak right here, bam, put them on the bench and squirt bacteria on one and smear it all over the top. A bacterium is a cell. It is alive. It has a metabolism. It has a way to take all 4 carbon compounds, break it down by different means, and that's what we'll discuss, to create ATP. If we came back in a couple days, there wouldn't really be much of steak left. Okay, they would have broken that down. We'd be left with a slurry. If we had on the other steak, instead, we squirt virus, the worst one you can think of, Ebola, measles, okay, all of that stuff, smear it on top of here. That steak is dead. Okay, we can agree to that. That steak has no metabolism. It can't do the work for the virus, and the virus itself has no metabolism for itself. So that later we would come back and there wouldn't be any extra or more virus. They would not have been able to reproduce and create energy from that. They're very, very different. So we start with metabolism to establish how it is that bacteria create energy, convert energy, convert energy, the different ways and how we can detect that they're there and, you know, identify them based on their products and all of that stuff. And this is basically what we're going to be doing in lab from here on out, is like, how do you know who it is? It's basically what is the end product of their metabolism. But with virus, they don't have one at all. And so we talk about virus in a completely different way, but it's nice to juxtapose them to bacteria. It is very common for me on the final exam to say, like, okay, your friend thinks that everything's a germ. What's the difference between a bacteria and a virus? I'm like, you know, and you got lots, I mean, you have a lot to say right now. Wait until you have 3 lectures on virus, you're gonna have a lot more to say. Okay? So that then it just is clear to you. You don't make those mistakes anymore. Those are very different microbes. Okay, very, very different. You could have started this class with a general idea, the other's different, but I'm not exactly sure if you will know. Okay, and be able to describe it to your patients. Okay? Alright, so the thing about bacteria, and this is why we have to do all of these metabolic tests in lab, and this is typically a lab question, is I'll ask, why are we doing all these metabolic tests in lab? Why do we do this stuff? It's because you can't tell them apart visually. There's only 3 shapes, and there's only 2 colors. And that's if they'll even take up the stain. Right? The gram staining, we use it all the time, blah, blah, but you know that there are limitations. Okay, organisms that refuse to take up the stain. Okay, if you won't take stain 1, you're pink in the end, right? So there's a limit to what we can be, what knowledge we can gain from a gram stain. And they're only coxy or they're bacillus, okay? Spiral, okay. And then they can arrange themselves as singles or diplos or tetrados for the coxy staphylos. But you still don't know who's who, okay? So that's why we have to do metabolic tests, because their differences are not structural. Their differences are metabolic, okay? That is one of the tripping points for students, because that's the exact opposite for humans. For humans, all of our differences are external. What you look like on the outside, that's how I can tell one person from another. Internally, you're all into one obligate arrow, that's it, whose end product is CO2 and water. So whereas in lab, we're going to be able to put microbes on different media and have the media change color or change pH and all kinds of stuff that's going to happen because of their different metabolic abilities. I can't do that with you guys. If I gave you all, I've said this in lab, I'm sure, gave you all a paper bag and I left the room and everyone breathed into the bag and I just like line them up here, like who's who, how am I supposed to know? Everybody has the exact same end product. Impossible. Our differences are external. Internally, we're the same. They're different, okay? They look the same on the outside, but they're very, very different on what they can do, okay? So that's a really key, important thought going forward. And so you're going to see a lot of unique processes that only they can do. And we already know that this is true because of those 5 tubes, eukaryotes are only in the first 2, right? Okay, obligate aerobe or facultative anaerobes, and those would be like yeast. That's a eukaryote. It's not human, but it's a eukaryote. What about obligate anaerobe? That's bacteria. Aerotolerant anaerobe, that's bacteria. Microaerophile, that's bacteria. They're doing stuff that eukaryotes can't, okay? So they have really unique fermentation processes. We're used to fermentation that produces lactic acid because we talk about lactobacillus a lot, and we're used to ethanol because that's what beer and wine uses yeast to do that. But there's all kinds of different end products, and we can test for those end products. And by finding them, know that this particular microbe is present. Okay? They do strange things like they'll do photosynthesis and not produce molecular oxygen on the other side of that. That sounds strange to us because that's not how plants do it, right? But that's just unique to them. And they can do what's known as lithotrophy, and that's using something inorganic as food. Okay, they can derive energy from that. That seems really strange to us because, for example, inorganic things are like salt. And you know that salt, okay, I like salt, all right, salt, whatever. I can't eat salt. If I go home and that's all there is, I gotta go to the store. Okay, or do without tonight because there's no such thing as I'm gonna eat salt. That's not a thing, right? You know that that's not food. It is to them. They can eat that. Okay? So that's strange. Not all of them, but some. And then methanogenesis. So this is, we blame the cows for the methane in the atmosphere. It's the cow. It's the bacteria in their intestine. Okay? Methanogens. All right. So I'm actually going to skip this because it just confuses the student. Although this is it right here, whether they're autotrophs or heterotrophs. So I'll talk about that. Let's say. We'll say any organism is an autotroph that can use CO2, which is right here in the atmosphere, and put it together to build macromolecules like proteins, lipids, carbohydrates, right? Nucleic acids. I can't do that. I'm living in the same exact world. I can't do that. I have to, right? If I need protein, I need to eat protein. Okay, like I can't eat sugar and hope for that's going to be turned into protein. That doesn't happen. Right? So I'm heterotroph. I have to eat something already built up. Okay? We are very complex organisms that in a sense, metabolically, we're kind of simple. I can't really do a lot of stuff. You know I already can't make folic acid. You know that part already. Right? And yet, like I need that, right, to do the other stuff, like nucleotides, right? But it's like I gotta eat that. Okay? So imagine being an organism that can just take everything that's right here and build everything you need. That's not us. Okay? We are heterotrophs. And a lot of bacteria are. How they put those carbons together is what this is about here. This is, like I said, this is the part that confuses students. Whoops, I didn't mean to do that fast. So we know that a metabolism has lots of different reactions within it, but most of them that we want to consider are building or breakdown. The building ones are anabolic. So if you can't remember, just try to remember how people get busted for anabolic steroids. Okay, that's for getting, like they're bulking up. Right? And so that's energy in. I call that an uphill reaction. That's building. Okay, that's anabolic. The opposite to break something down is catabolic. Right? And so that's energy comes out of the system. I call it a downhill reaction. And so before, I've shown this same image before, and just to remind us, if this is a protein, then these boxes are what? Amino acids, that's right. And if this is DNA, then what are these boxes? Like which one? A, T, C, G. Right, exactly. That's a generic picture that means the same thing, okay? So we recognize that the big macromolecules that we utilize all the time before carbon compounds are themselves built up in smaller products. Okay. So there is a little conversation about free energy, which I'm going to go very quickly over. Built things have energy in them. The bigger it is, the more energy is in it. And so we can just say that, blah, blah, blah. Okay, what does this mean? Okay, the energy in any molecule, okay, so let's consider molecules because we're not working with kinetic energy like the guy jumped them. Okay, that's a molecule. We can agree with that. Okay, what is this? Okay, and what is it?
Sharing electrons.
Electrons, that's right. Okay, and what is an electron? Okay, so then it just kind of gets a little bit kind of nebulous. But basically, you've probably heard that it has almost no mass. It's mostly just energy. So the bigger the molecule, the more electrons holding it together, yes, the more energy it contains. Okay, so big molecules have a lot of energy. Small molecules have less energy. Okay, so when we go from this is glucose and break it down into CO2, a lot of energy has been released. Okay, that energy is actually the electron. And it's the electron that's captured and used to create ATP. That's this whole conversation right there in a nutshell. Okay? So, things are more stable in a low energy state. Okay? So we can sort of understand that, and so let's just pretend that this is not a grown person, but a 2-year-old, and now we understand very clearly that one is less stable than the other. Okay, maybe not in water, but okay. Somewhere safe. All right, but like up high, if I have a bunch of bowling balls up high, that's less stable than if they were on the ground. Yes? Okay. So same thing. Big built up stuff has a lot of energy, but it's actually more stable for it to be broken apart. Okay? And we know that when something is broken apart, now it's disordered. That's called entropy. And you might have heard that entropy is always increasing. Disorder is always increasing. Order takes energy to keep it that way. Okay? So you can have like a conversation about
anything you want, society, anything. It takes energy to keep order. Disorder, that's the easy part. Okay, that's what the world wants. Break it down. Crush it. Okay, it takes energy to keep built things. Think about yourself. You're a huge built thing. Okay, you're a huge built thing with a certain temperature that you require because you have an optimum temperature. Yes. Okay, and this room is not your temperature, so you're in a fight right now. You're not equilibrating, right? We've established that already. Okay, so we just recognize this, that built stuff takes energy to maintain. This is the easiest. For catabolic reactions, they're exergonic, that's breakdown. Okay, we're releasing energy. Remember that the released energy, when you go from having all of these carbons bound together to not bound together, means that it's the electrons that are released, and the electrons are the energy. Okay, the energy is released in the form of electrons. It's the opposite to build something. You have to put energy in. And so if I want to put two carbons together, there have to be electrons that are shared. So electrons have to be added. Okay. So for the cell, they have to break down materials. Glucose is everybody's favorite. Okay. Glucose is a sugar, 6-carbon molecule, and it gets broken down in metabolism. Okay. There's different ways to do it: glycolysis, Krebs cycle, respiratory chain, and also fermentation. All of this yields energy. Okay, we break it from a 6-carbon thing into a 3-carbon molecule of pyruvate, and then what? Then you use the energy from here to build everything that the cell needs: building blocks, macromolecules, all of the rest of it. And we know that all cells are powered by ATP. So I haven't really been saying anything yet, have I? Right? This is just sort of laying the foundation for what we're going to talk about. And so this is ATP, and you've seen it before. And so this is a ribose and a amine group, and then 3 phosphates, and we have this squiggly line here because we know that in solution the hydrogens are lost, and I have 3 negative charges next to each other, so we have repulsion, charge repulsion between the phosphates. Okay? And so when that phosphate is released through hydrolysis, remember when we built anything, it was dehydration synthesis, take the water out, add water back, breaks it apart. This phosphate that is released, we have some energy that is released with it. What's really released is that line, and that line we already said is the electrons. Right, so that's what's released. And so we have ATP becomes ATP and a phosphate. ATP is basically how you pay for everything. So the ATP gets broken down into ADP and a phosphate, and that energy is used for something that requires energy, like building proteins, building nucleic acids, all of that stuff. And so how do you get this back to being that? And that is what the metabolism conversation is about. Okay, so we'll spend a little bit of time on this picture, and you should be able to recognize it. On the metabolism worksheet, I have this image. And so we are in microbiology, and so we are talking about the production of ATP, which you know for bacteria on the plasma membrane. Okay? Above that, and they've chosen to show you a gram-positive, what is that thing above the plasma membrane? That's the cell wall, right? That's the cell wall. Okay. All right, so now here, this is an electron transport chain. Okay. This is oxidative phosphorylation, the electron transport chain. We've done this before, this is review. Okay.
That's one way to make ATP, but the other way is substrate level phosphorylation, and that means enzyme. So you know that enzymes work on substrates. Okay? And there are some enzymes that can take a molecule that has a phosphate on it and pull it off of that molecule and add it to ADP to become ATP. Those are the only two ways to make ATP. I can have fancy names for the processes, but it's only two ways. You're either doing it substrate level or you're doing it oxidative phosphorylation. Okay? Oxidative phosphorylation is the electron transport chain. You will see that when we talk about glycolysis, we make 2 ATP, substrate level. When we talk about Krebs cycle, we make 2 more ATP, substrate level. When we talk about fermentation, we make ATP, substrate level. The other way is the electron transport chain, that's oxidative phosphorylation. So don't get yourself all tied up in the names of these processes, because like, which way is it? Enzymes are proteins. I love proteins, you know I do. But enzymes can only do one reaction at a time. One. So when you see single-digit ATP production, you're looking at substrate level. Because electron transport chain, as we see over there, if you're using molecular oxygen as the final electron acceptor, you can bat out 38 total. Bam. Okay. 36. One of the things that we have to spend some time on is this NADH right here. NADH is a molecule that is an electron carrier. So when we say that we're going to break apart glucose, so take the 6 carbons, so just think about those pop beads, those, we're going to pop them off so that they're all singles. Well, what happens to the electrons? NADH will take those electrons, but it doesn't want them. And you'll see why in a moment. So when NADH is near the first protein of the electron transport chain, that protein is very electrophilic. What does that mean? Likes the electron. It takes the electron from NADH. The protein next to it is even more electrophilic. Steals it from that one. The one next to it is more electrophilic, steals it from that one. Okay? All the way, and I drew this myself so I couldn't do the curve properly. It should go all the way across. And then it will be added to a very electrophilic molecule, molecular oxygen. Okay? Very electrophilic. As we pull electrons in this direction to our final electron acceptor, we also take the hydrogen here and pump it up. Okay? Hydrogen ions, they're really small. Think about hydrogen on the periodic table. It's the smallest element there is. All it is 1 proton and 1 electron. And if it doesn't even have the electron, then it's just a single proton. Okay? Very small, but it can't slip through back into the cytoplasm. Why not? It's charged, so it's a problem. What's keeping it from slipping back? Or what about the plasma membrane? The lipids, because they are nonpolar, and they don't like that at all, repel. But we have this situation where, let's pretend these are in fact bowling balls. One after another, and I just keep putting it on this shelf. Does that seem stable to you? No. So it's got to come through, but it comes through a specialized pore known as an ATP synthase, which is an enzyme. Okay, ATP synthase. They flow through ATP synthase just like water flows through a water wheel. And ATP synthase takes ADP, and if you have this drawn out or whatever, do plus a phosphate, because it can't just suddenly be ATP. Where's the phosphate? Okay, ADP plus a phosphate becomes ATP.
So this ATP synthase crimps together ADP and a phosphate and creates a TP. So we'll have a little conversation about enzymes in a couple of slides. Enzymes love substrate, love substrate, and have such great affinity for substrate, none for product. So it wants to crimp together ADP and a phosphate, but as soon as it makes ATP, spits it right up. So its mouth is wide open to do it again and again and again. The more hydrogen ion that you have up here, the more ATP that is made. The best way to do this system, the electron transport chain, is to use molecular oxygen as your final electron acceptor. That's what we do, and we make a ton of ATP by doing it this way. The problem is, let's think about molecular oxygen. You know that it's an electron. Do you think that molecular oxygen is waiting politely for its turn at the electron? No. No. So it's taking it wherever it can. And if molecular oxygen gets an extra electron, we have a special name for that. What is it? Superoxide. Well, that's really reactive. What are you going to do about it? What enzyme? Superoxide dismutase. Superoxide SOD. And catalase. That's the whole story about those 5 tubes. Is all like, do you have it or you don't? Right? So those organisms that utilize molecular oxygen have superoxide dismutase, yes? Okay, because they know this is happening, but they can stomp it out. Boom, boom. Right? They can stamp it out. What if you don't have those enzymes? Okay, well, you can use something else as your final electron acceptor. Nitrate. You wind up with nitrate as a final product. Sulfate, you wind up with hydrogen sulfide as the final electron acceptor. Okay? Why wouldn't you want to use oxygen? It's the best one. What if you can't control it? Then it's not the best one for you at all. It's actually toxic. Right? So they use something else. Well, let's talk about these two. Are they as electrophilic as molecular oxygen? No. So are they pulling on the electron as much? No. And if you're not doing that, then you're not making as much hydrogen ion gradient. So you have, instead of a big rush of water through your ATP synthase, you have a trickle. And you make a little bit of ATP. And that's going to show up here when you're looking at the overall growth. Tons for the obligate arrow at the top. A few for the obligate anaerobe at the bottom. Because unlike us, they know exactly what they're here to do. Divide, copy, more. And they would sure love to make more of themselves, but they just don't have the ATP for it. Because they choose to use something other than molecular oxygen in the electron transport chain. So, if you're using molecular oxygen, you say you're doing aerobic respiration. If you're doing the electron transport chain, but your final electron acceptor is something else, that's anaerobic respiration, which I recognize sounds like jumbo shrimp. Like, how can this be? Anaerobic respiration, but there is such a thing. Wow, that's so weird. Yeah, it sure is. Only bacteria know how to do this. So let me just, I said it several times, Google, not your friend. You look up anaerobic respiration, it's going to tell you all about fermentation. And you're like, that's a different process. It doesn't even involve the electron transport chain. It's something completely different, substrate level only. Like, suddenly you know more than Google. Okay? You recognize that Google is the human construct, like here's all the known stuff and dump it in, and whoever put this together isn't a microbiology student. Right? So you would have added that. Like, that's a huge world. Hello? They outnumber us by many. Right? How come they're not in there? I don't know. We know about them. Okay, so just be careful because Google will send you down the wrong. Okay? Wait till we get to our unknown. And you're gonna have to tell me what kind of metabolic type they have, and you type it into Google, and you do a test, and you'll show me that it has no catalase. If it has no catalase but can grow on the surface of the plate, that's an aerotolerant anaerobe, and we'll prove it to ourselves by, yes, they don't have catalase, yes, they have no catalase. But you'll type in the name of the organism into Google, and it's telling you facultative anaerobe. Red mark. No, you proved it to yourself already. Don't let Google tell you what's going on. Okay, be careful here. Google's great for other things, you know, how to get to some place. That's what they're, I mean, right? And even then, right? This is a very important slide. How long have I been talking about this? Okay, so this is our third unit together. Now you know. Gosh, she was there for like 10 minutes. It's a really important slide. All kinds of stuff comes out of here. There's more than just these two as choices for anaerobic respiration. Okay? And each kind of final electron acceptor will yield a different end product. I can test for that. Then I'm going to know who I'm dealing with. Okay, if I'm testing for, does this organism make water? Well, I know it's an anaerobe. I'm not even sure if it's a human, right? That's not telling me much. It's this stuff that gets me. Okay.
So,
You had a question.
yeah, just to clarify, so the difference between aerobic and anaerobic is just the end product, basically?
No, the final electron acceptor,
The final one.
which is what for the aerobe?
For the aerobic, it's going to be oxygen.
Okay, and for the anaerobe,
NO2 or
yeah. And the other one that we'll think about is CO2, which the end product would be methane, CH4.
But that's still oxidative phosphorus.
Yes, it is, because it's using an electron transport chain.
Okay.
So the reason why it's called oxidative, one, yes, they figured it out with oxygen first, but it also has to do with oxidation. The term oxidation, right, loss of electron, we're all losing. Okay, but what about oxygen? Did it lose an electron or gain it? Gained it, and what is that called? Reduction. That's it. That's actually really a very simple process, oxidation and reduction, but I don't know what it is about, probably, I hate to say it, I'm not trying to diss your chem instructor, but I'll bet you came out of there completely confused. You had no idea what oxidation and reduction were about, when they're used, what is the importance, and yet here we are, and this is what we're gonna be talking about, that's it. And there's no mystery here. I don't know why it's muddied so much. So, if I have done nothing more for you than give you the silly phrase, Leo says, girl, take it with you. Okay, that will keep you where you're supposed to be. All right. So, for any reaction to occur, we know that we have collision theory to think about. So, reactions take place when molecules collide in the correct orientation. So let's just pretend that this is the reaction. I want the cap on the pen. Okay? So they have to collide in the correct orientation. This is not working, right? Has to be in the correct orientation. All right? And with enough velocity, because okay, like that, it's not gonna work. It's gotta go a little faster, okay? And so concentration will help a lot. And I'm showing you this reaction with my hands. But what if I had the pen and the cap in a bag? Okay, and I want the cap on the bag, then I have to shake. Okay, I'm going to shake it. And I'm going to, have some level of success. It's very low. But what if I had 100 caps and 100 pens, and I shook that? I have just increased the likelihood of my reaction happening. So concentration is important in reactions as well. The amount of energy required to put into a system to get a reaction, even as simple as putting the cap on the pen, imagine how hard I would have to actually do this. That's a lot of energy to get to that point. And yes, I can increase my likelihood by putting 100 caps and 100 pens, but I still have to put in a ton of energy, right? the reactants are at a lower lower energy state. But we in all living systems use enzymes. Enzymes are catalysts. They make a reaction happen faster than it would otherwise. So the bag is one way to get that reaction, but a really simple way is to have my hands be the enzymes. If my hand is the enzyme, then it's happening correctly each and every time, and I'm done, and I can do it again, and I can do it again. My hands don't get used up in the reaction, right? So these are some of the things. Now the problem with this, if you're looking and you can really consider how hard would I have to shake that bag to get that reaction to occur, I mean, I'd be going crazy over here versus almost no energy at all. So unfortunately, we're looking at this cartoon image that is not giving us this idea. It seems like it makes it a little bit faster. It makes it a ton faster, a ton faster. In fact, if I were to draw what would happen with the energy without an enzyme, that curve would be way up there, okay? And the enzyme makes it happen with a whole lot less energy involved. Yes?
Why are the products at a lower energy state?
So
Is
the
that what you
product,
said?
yes.
Yeah. Wow.
So why? Because they are, so this has to do with the Gibbs free energy. So how much energy is involved in the system, and that these products want to be in this state.
Okay.
Okay? That's not a satisfactory answer. I get that.
Yeah.
Right, okay. I want us to think that the enzyme can do the reaction with a whole lot less energy involved than without. If you think about like having a hamburger, right? And if I were to leave the hamburger out, we know that entropy is always increasing. The hamburger would in fact break down. But if you eat it and you have enzymes, as you do, okay, the breakdown process, this has been estimated to take on the order of 200 years. But you eat a burger and you're ready for something else in like 2 hours because you have enzymes. That's a huge difference, 2 hours to 200 years. So enzymes are absolutely key and necessary for living systems because otherwise nothing would ever happen. Okay? So that's what I want us to recognize. So they're lowering the energy of activation. And I almost always ask this question, why and how do the enzymes lower the energy of activation? So they do speed up the reaction and they have high reaction rates, but the reason why they lower the energy of activation is because they put the reactants in the correct orientation so that they're facing each other in the right way. Okay. And because of that, putting it not only in the right way, but also what's called stabilizing the transition state. So think about this reaction here. Okay, the cap's on there, but you know there's that final little oomph to actually get it on, okay? Because if you just stick it on here and flip it over, you know this is just gonna drop, right? So that final little oomph, that's called stabilizing the transition state. So orienting the substrates and stabilizing the transition state is how the enzyme lowers the energy of activation. Right, that's how it does it. And because they're not involved in the actual reaction, they can do it again and again. That's the high catalytic turnover rate. Okay? So some enzymes require cofactors, and we talked about that today in lab. We said that DNases in the cell, okay, would chop up DNA, but they need magnesium to do it. And that's why the instagene mix had a chelator to bind up all the magnesium so that the DNase can't work. Okay, so a lot of enzymes require cofactors. They're either vitamins or minerals. That's what they're doing for us in our diet, is we need them so that our enzymes work properly. Okay? They are affected by inhibitors, and we know this, competitive and non-competitive. A competitive inhibitor does what? Blocks the active site. A non-competitive inhibitor binds somewhere else on the enzyme but changes the shape just enough that it doesn't work properly either. We have all kinds of examples of competitive inhibitors. Penicillin, amoxicillin, rifampin, quinolones. They're all working the same way. Trimethoprim, sulfamethoxazole, they're all doing the exact same thing. So it's like we learned about that before we really learned about enzymes. Okay? But we now know that. They can certainly be affected by that. And because they're proteins, they're really requiring their shape for their function. So anything that would mess up the shape will mess up the enzyme. High temperature, pH changes, right? That would break hydrogen bonds and then change the shape. So pH changes, we use these. This is just an example from our system, from our digestion. And we have different enzymes in digestion that take part in different places. You know that your mouth is mostly neutral, and you have salivary amylase. It works optimally at 7. That's the pH of the mouth, roughly. Okay? But you swallow, you chew and you swallow, and it goes down to the stomach. The pH of the stomach is 1.2 to 3. Okay, so lower when you're younger and higher when you're older. But at 1.2, look what happens to amylase. The amylase activity is absolutely non-existent at pH 1. This is over. There's no more carbohydrate digestion in the stomach. Okay? But in the stomach, you know that the cells that line the stomach squirt out pepsin, right, and hydrochloric acid, and that's the low pH. That's where pepsin works. Pepsin loves pH 2. Look at that. That's its max. Right? You leave the stomach, the acid kind gets squirted into the duodenum. Okay? And immediately the pancreas squirts in to raise the pH to 8. Pepsin's dead at 5. There's no pepsin activity at 8. But here's who is working. Arginase and lots of other enzymes work there too. So this is an easy way to kind of flip off enzymatic function is a pH change. We do this in our bodies. Okay? This is just a general checklist for enzymes, and typically these questions about enzymes, now I will ask how does it lower the energy of activation? First off, you have to know what the energy of activation is. This is how much energy it takes to make a reaction happen. Okay, and I showed you a building reaction, but breakdown reactions are also reactions, so they're pulling apart. Okay? Most of them are proteins, many require cofactors. Okay, lower the energy of activation. Unique characteristics, right? What did I want to say? So this one. Associate closely with substrate. So they love substrate, but have no interest in the product. This is why the active site is ready immediately for another round. Because it has no interest in the product. It only wants the substrate. Once it's turned it into the product, it spits it out, it's ready to go again. Over and over and over. Okay? So, the energy of activation question will be asked, right? What is it? How is it that enzymes lower this energy, orienting the substrate, stabilizing the transition state? Everything else about enzymes will be presented as a multiple choice. All the following are true, except, or none of these are true, except, or whatever. Okay? So it could be a couple questions on that. So it's not like you have to memorize everything, but just kind of know what's true about them and what isn't. Okay. So you've seen feedback inhibition or heard about feedback inhibition before. And so proteins enzymes, there's lots of different protein functions. Catalysis is just one of the 7. But this is a protein, this enzyme is a protein, its information lies on the DNA, right? Okay, and so DNA gets transcribed into message, gets translated into protein, which folds and does its reaction. In cells, the reaction creates products, and sometimes the products go over here and block the gene for that enzyme. We've already made enough, that's enough, stop making this. That's feedback inhibition. So if it sits on the gene, then RNA polymerase can't copy it into message, and we don't make any more of that. Okay? So that's just a feedback inhibition. Some enzymes are going to work inside of the cell, and some are going to be secreted. We have all kinds of secreted enzymes. Pepsin is one. Bless you. Pepsin is one, right? So it's sorted out into the bag that is the stomach. Okay? Amylase, sorted out from the epithelia lining the mouth into the mouth cavity, right? So we do this a lot. Bacteria do this too. Okay, so we'll be checking for catalase and coagulases and all kinds of stuff. These are really secreted enzymes. Excellent. And so this is just showing you that energy is 2 kinds, either kinetic or potential energy. This is potential up here. Kinetic is actually the energy of motion, but we can't make any use of that. So a really hot room is not a way for me to make ATP. I don't know what to do about that. Okay, the brightest light ever, I can't make ATP out of that. So we just can't harness kinetic energy. We need potential energy, which is chemicals. Yes, what is your question?
So for exoenzymes and endoenzymes, does that mean one secreted outside?
Yes. And one
I'm
works
assuming
within the cell.
endoenzymes are the ones secreted.
They're just made inside of the cell, and they stay inside the cell. Okay, so molecules have potential energy. I already told you about this, but energy is here in the bond. Okay, that's the electrons. And when we break down materials like glucose, we're basically taking away all those bonds. Okay, all of those bonds are releasing the energy every time we break a bond. And so we are going to use those electrons, I already told you how, to make ATP. We break down the glucose into 6 of these 6 parts. And so whenever you're talking about the moving of electrons, you're just talking oxidation reduction. And so I will show you the picture of the electron transport chain, and I'm going to ask you who is getting reduced and who is getting oxidized, and it's the simplest thing ever, unless you haven't taken the moment to remember Leo says okay? If you're losing an electron, that's oxidation. If you're gaining it, it's reduction. It's as simple as that. Okay, that's all it is. So if you're saying that glucose is losing its electrons, then it is being oxidized. Okay, but who's gaining them? Who's my final electron acceptor?
Oxygen?
Oxygen, molecular oxygen, is reduced. Is that what you meant? That was what you were trying to say. Okay. Yes. So what is actually carrying my electrons is NADH. Okay. Here's NADH and here's NAD+. And this is actually a much bigger molecule, but this is the business end of this. Okay. This screwed me up for years and years and years as an undergrad and in the beginning part of my graduate studies as well, because all I ever saw was NAD+ and NADH. And it never really made sense to me until I looked at the structure, and then suddenly everything clicked into place. Very, very simple. Okay. So what we have here, let me just do this. And I know you love when I draw on paper. I'm kidding, I'm sure it's alright, but alright, so NAD+, NADH. double, so this is a ring, okay? And so here's a double bond, skip, double bond, skip, double bond, yes? Okay. And sometimes in chemistry, like that would be written, where can I do this? I guess here. Okay, so at this point we have, oh, I have this. Alright, so we have here, here, and here, yes? And then it's often drawn like this. Here, skip, here, skip. And these are equivalent, yes? Okay, this is called resonance. You've heard this? You've not heard of resonance. Okay, well, what this means is, well, what are these two lines? What does that mean? What is that? It's a, wait, what is that?
The sharing of 4
yeah,
electrons.
Shared electrons, right? Okay, so what's going on here is that you've got 2 electrons, which we said is just energy, right? And so when you have resonance, this ability for anybody in this ring to be the double bond holder at the moment, and this happens like instantaneously, this is more stable for the ring as a whole, right? For everybody to be hanging on to this very energetic electron, right? Electron. Okay, that's all well and good. This is NAD plus. NADH is so close, except that if you look at this carbon here, this carbon can only ever make how many bonds? 4. So 1, 2 to stay in the ring, and now it's doing resonance, so 3, and here's my 4. Okay. NADH, that carbon now has 2 hydrogens bound to it. It can only ever make how many bonds? 4. So now 2 to stay in the ring, it cannot play resonance. It's like, sorry guys, I can't do it. That's on you guys. So that forces these 2 sets of carbons here to always hold the energy. That's less stable. What do molecules want? More or less stability? More. So NADH, and I like to write it like this so I remember what the heck's going on here. Okay, that's the hydrogen right there. That's the 2 electrons. I call NADH the reluctant electron carrier. It doesn't want them. It doesn't want them. It wants to be NAD plus so that it can do resonance and be more stable. Okay, so this is less stable, and this is more stable. Okay? It doesn't want that. So as soon as it gets near this, it's like, thank you. Phew. Okay? But it's not just the electrons that are being passed. It's also the hydrogen. Right? Okay? And now it's back to this. Yes?
What's going on with the nitrogen?
Oh, nothing really. I mean, nitrogen is really electrophilic as well, but it will just...
I just noticed one had 2 dots above it.
Yeah, well, so, yes, it's got a lone pair there, but it's like over here it doesn't because we have the resonance kind of moving those electrons all over the place. Yeah. This is the less stable, this is the more stable. Okay, so NADH, less stable, it acts as a hydrogen and electron shuttle. And so here I'm sorry. This is what it really looks like a really big sort of ribose with the adenine and all that business, but that part's not even taking part in the actual reaction. It's just up here, this ring. And so what ends up happening is that this, as we said, is going to give up its electrons. Where is my picture? Here. Right there. That's what happens. Thank you, take it away. I don't want them. Okay, great. So it is the shuttle for electrons. And what's happening is, let me just jump forward for a second. In glycolysis, where you have a 6-carbon molecule, I'll raise it here. Where you have a 6-carbon molecule. Okay, so they don't like me to draw up there. So I'm gonna do this here. So 1, 5, 6. You know, glycolysis is a 10-step process with, and each one of the steps is an enzyme. Okay, and at the end, this is all you managed to do. Is to make 2 pyruvates. So what actually happened? Right, so these are gone. What is that? What is that? 2 electrons. Guess who took them? NAD plus and is now NADH. Does it want them?
No.
No. Okay, so now we take the pyruvate. Let's just follow the fate of this one. Before it goes into the Krebs cycle, it does a CO2, so you just have 2 carbons that go into the Krebs cycle. That means this is gone. Guess who took them? NADH. Okay. And now these 2 carbons, they get peeled off too. Right, so it just has single carbons now. And guess who took this? Yeah, NADH. Okay, and does it want them?
No.
No. And so it goes over to the electron transfer chain and just dumps it. Now the thing is, we have to learn stories in glycolysis and electron. It's like, okay, NADH doesn't want this, and that means it doesn't want them right this second. So as soon as it's happening, it is yielding them to the electron transport chain. It's as soon as, okay? We can't learn things like that. But that's how it is. So,
It's not waiting for press to be taken again.
right, it's like it's happening at once. All right, so it's dumping them all off and we're making ATP. Okay. So that's glycolysis, Krebs, the electron transport chain. And so if you use oxygen as your final electron acceptor, that's aerobic. But if you use something else, that's anaerobic. Okay. So each of those will have a different end product. It's not going to be water, okay? It'll be nitrate, hydrogen sulfide gas, or methane. That's different from water. I can detect that. Okay? So that's how I know who's there and what's going on. The thing is, these yield less ATP. Okay. This I'm going to do. I don't. Okay. So these are our different metabolic types. I ask for the metabolic type in the unknown. I will ask for it. I just did in the exam the other day, and I asked for it also on this lab exam that's going to come up. The metabolic type means which tube is it in? We're not used to that and having to describe the metabolic type because everybody's the same. You're an arrow, big deal, right? Like, we're not talking about that. And what we say is like, I have Heimtaubes. I have whatever arrow, okay? Like, that's not right. Like, you're like, you have to be specific.
And what I do know is that if you're using molecular oxygen, you're going to make max ATP, and I'm going to detect that in the number of cells produced, because that's what they use their ATP for. Okay? Do you see how few there are down here? Okay. Why? Because they don't have enough ATP. Why? Because they're using something other than molecular oxygen as the final electron acceptor. Why? Because they can't control what happens when you use molecular oxygen. It can't help itself. It's an electron hog. It steals electrons even from the molecular oxygen right next to it to become superoxide. That's what you're dealing with when you're dealing with molecular oxygen. We're not used to it. We're thinking oxygen's your friend. It kind of is. It is going to win in the end. It always does. Right? Ask the pail in the backyard. All right. So these are the different kinds of metabolic types. And I told you before that up here, the facultative anaerobe will run an electron transport chain. It's the best way to make ATP. But what if there isn't any molecular oxygen? Well, for the aerobe, that means you're dead. Okay, that's what would happen to us. I only have 2 minutes without oxygen. I don't have a second plan. But for facultative anaerobes, they do. Their plan is to ferment. Okay, aerotolerant anaerobes only ferment. Okay, and then these guys are running an electron transport. Okay. This is what we're looking at when you're talking about bacteria. This is happening either in the cytoplasm or in the membrane, because guess what? That's all there is. There isn't some other secret compartment inside there. When I show the image of the electron transport chain and I ask in the question, where is this happening in bacteria? And the in bacteria part is bold and underlined, and I get mitochondria as the answer, you get the very sad face. This is unit 3. We can't still be talking like that. Okay? Yeah, that happens in mitochondria, but if I'm asking, what about bacteria? Well, guess what invented this whole thing in the first place? All eukaryotes did was like, wow, that's a great system. Let's swallow that thing. Okay, right? That's what happened there. Okay. So it's not just sugars that are used in metabolism. This is just showing you that other foodstuffs, of course, are part of it. They're just going to come into different parts. However, I want to spend one moment on this. The use of proteins, and we need them, right? We consume them all the time, always leads to an amine group released. Amine groups, so for us, what we do is we take 2 amines, we slap them on either side of a carbon and call it urea, and we get rid of it in our urine and in our breath and in your sweat, right? It's just out there. But amines are basic. And so if I have a pH indicator in my media, in my plate, or in my broth, I can see a color change. And that's differential. Okay, so if I see it go, ooh, that's basic, they're eating protein. Okay, then I know. Okay. Anaerobic respiration, as I said, will yield different final end products, and this one shows the CO2, that's why I showed that there. But that's not the only thing. Okay, at the end of glycolysis, you're either going Krebs cycle and an electron transport chain with molecular oxygen or something else as your final electron acceptor, okay, fine. But there's an entirely different thing, and that's fermentation. Fermentation, do you notice, does not use the Krebs cycle, does not use the electron transport chain. It's its own thing over here. Okay? So these are the 3 ways to make ATP. Aerobically, okay, glycolysis, Krebs, electron transport chain with molecular oxygen as the final electron acceptor. Anaerobically, glycolysis, Krebs cycle, and the electron transport chain, but then, you're using something else as the final electron acceptor, or you can ferment. Look at the ATP yields. This is max, right? This is less. Why can't I give you a straight answer? Because, well, which one are you using? What is your final electron acceptor? Then I will tell you what your final ATP yield is, okay? So all we need to consider is it's less than molecular oxygen. That's good enough for us. But you notice that here we just get 2, but the end products are what? Alcohols and acids, you've heard this before. That's the end product of fermentation. I can detect for that. I can know if an acid is made if I have a pH indicator. Right? That's easy. So, fermentation is going to take the pyruvate, which is a 3-carbon molecule, right? And the entire purpose of fermentation is to regenerate NAD+. So that means take the NADH that was made in glycolysis and take the electrons away from it. Okay? Those electrons are given to an organic molecule as the final electron acceptor. That organic molecule is an acid or it's an alcohol. What does it mean if something's organic?
It's carbon.
How many? Is CO2 organic?
Yeah. No, because it has a free,
Two. Just 2. That's it. Just 2. Okay, so that's the final electron acceptor is something organic, 2 or more carbons. Now, we say that aerobic respiration is doing the max ATP yield because you're getting all the electrons off. Pop, 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 pop. You're popping off all those carbons, releasing them as CO2. But if in fermentation, you're leaving me with an acid or an alcohol that still has 2 or 3 carbons left, there's still energy left in this system. You have not torn it down to the studs. So it's less efficient, okay? It's less efficient. And so guess what? If you are a facultative anaerobe, well, why should I ferment if there's oxygen around? I could make 30, what would you rather have? 38 ATP or 2? 38. Obviously. So if you are, if you are talking about yeast, beer and wine, the first thing you have to do is get rid of all the oxygen. Otherwise they won't do it. They will not do it. Okay? That's true for the facultative anaerobe, but if you're aerotolerant, you're going to do it no matter what. And so that's where another place where Google, not your friend, okay, because it doesn't require oxygen, but will happen whether oxygen is present or not if you're an aerotolerant anaerobe. Okay? Aerotolerant anaerobes ferment all the time whether oxygen is present or not. That's not true for the facultative anaerobe. That's different. So the electron transfer, they don't even own it. They're not doing that. They're fermenting. So Google will tell you that fermentation is a process to make ATP when oxygen is not present. And it's like, that's not always true, is it? Google, learn about aerotolerant anaerobes, please. Right? All of you who know somebody who work at Google, fix that. That's a serious problem. All right, so here, you will know one organism and its unique organic end product from fermentation. So, let's look at a couple of these. Now we know Streptococcus, Chromophilus, Lactobacillus, they make lactic acid. We know that. Well, so does Bacillus. They can do it too. Okay? Pyruvate is a 3-carbon molecule. Lactic acid is a 3-carbon molecule. Okay, so what's changed? It's just a little bit of working around, taking the electron off of NADH to become NAD+. Okay, fine. Yeast do this too. Yeast is off the table. Why? Eukaryotes. Not you. Okay? Lactic acid is a 3-carbon molecule. Remember I told you there's still energy left in the system because there's still bonds there. Propionic acid bacterium, sometimes called propionibacterium, will take that lactic acid and they pull off one of the carbons and release it as CO2. And they make propionic acid and acetic acid, that's vinegar. These two organisms together, lactobacillus and propionic acid bacterium, are used to make Swiss cheese. That's the hole in the Swiss cheese, is the CO2 release, right? And then hydrogen gas and the acetic acid gives it a little bit of, acidic flavor. Not everybody likes Swiss cheese. Okay. Clostridium, right? Now, not every kind of Clostridium is an anaerobe. They can also ferment. Look at some of these end products. Butyric acid, butanol, acetone. Acetone, I use that to take off, nail polish, right? Except that it's a known carcinogen, so please limit your use of acetone, okay? Isopropyl alcohol, CO2. That's a lot, okay? On this list here, let's decide which ones are and are not organic. Butyric acid. Organic or not? Okay, I'll do the talking. Okay. organic, inorganic. Okay, CO2 cannot be your what gives me one organism and a organic end product. You can't say CO2. Okay, pick something else. Acetone, butanediol, whatever else. Okay. All right. E. coli and salmonella make ethanol, lactic acid, succinic acid, plus acetic acid, CO2, and hydrogen gas. That's a lot of acids. This is going to come into play in lab. We're going to know E. coli's there if there's a ton of acids on the other end. They're excellent at this. E. coli, remember, are facultative anaerobes. I showed you an actual image of them. And they're very good at it. They're growing all the way to the bottom of the tube. Right? They prefer oxygen, absolutely. No oxygen, no problem. They're just gonna have it. Right? Okay. Enterobacter will be another organism that we look at in lab, and they're very similar to E. coli. They're both gram-negative bacilli. I can't tell them apart on a gram stain, but when we test them for what is the acid production, this one, ethanol, well, that's not an acid. Lactic acid, they do the same. Formic acid, that is an acid. Butanediol, that's not an acid. Acetone, that's not an acid. CO2 and H2, well, there's more acids over here. I can tell the difference based on do you make a ton of acid or not so much. That's how I know which organism it is. Not by looking at it, because they're both going to be gram-negative bacilli. That will leave me scratching my head. I don't know who's who. I have to look at organic end products. We do use a lot of fermentation end products. Like I said, acetone and isopropanol are in fact made using Clostridium, E. coli and Acinetobacter for acetic acids and vinegars. And as I already described, Swiss cheese may be already too. Okay. So I asked for one organism and one organic end product. Here's who's off the table: lactobacillus and lactic acid. Because you already knew about that, and Saccharomyces, that is a eukaryote. Okay, so here are other organisms. So Klebsiella, we worked with that before. Okay, if you make a lot of sense. 2, 3, 4, 2 down. Your choice. This is exactly what lab's going to look like for us. We will start with a tube that looks like this. We will inoculate with all different organisms, and we come back later and like, wow, they're all different. Yes, because they're all making different end products, and that's how we're going to be able to tell them apart. Okay? We'll be able to tell fermentation on plates like this, and then this very shiny thing, this is part of the coliform analysis. Right? E. coli will look like that, with no other. Okay? So this is how we tell apart bacteria that are otherwise structurally similar. When I see a gram-negative bacilli, here's what I know. It's not a gram-positive cocci. So I can eliminate all those, but I don't know who it is amongst these. Right? Then I gotta continue with the lecture. So this is our unknown. All right, we'll stop.